Welcome to the deep dive. Today, our mission is really to shortcut your understanding of something pretty revolutionary in biology. The idea that you're not just you're a walking, talking ecosystem.
Exactly. And we're diving right into that ecosystem with a situation that honestly drives home just how much we rely on our microbial partners. Think about severe recurrent clostridioids, difficile infection, C. diff for short.
Right.
We mean chronic diarrhea, serious weight loss, people basically confined to their homes, and often it kicks off after just a standard course of antibiotics.
That sounds absolutely awful. Before we jump to the radical cure you mentioned,
Yeah.
we need to understand the bug itself. How does C. diff, which you said is often normally there in small numbers, how does it survive the antibiotics that wipe out everything else?
Yeah, that's really the key point. C. diff is a gram-positive bacterium, a firmicute, but the crucial thing, it forms endospores.
Ah, the spores.
Exactly. These spores are incredibly tough, resistant to cleaning agents, many drugs. So when broad-spectrum antibiotics come in and clear out the normal gut microbes, basically, it's competition these dormant C. diff spores can germinate.
And then they take over.
They overgrow, release powerful toxins, and that's what causes that terrible, debilitating diarrhea. It's a problem of opportunity.
And when antibiotics just don't work anymore, maybe they trigger another round of C. diff or the strain is just resistant, that's when doctors have turned to something called fecal microbiome transplant, or FMT.
It sounds radical, but it's actually quite elegant. You take feces from a very carefully screened, healthy donor, process it, mix it with sterile saline,
Okay.
and then infuse it into the patient, usually through a nasogastric tube or sometimes a colonoscopy.
The success rates are, well, they're kind of mind-blowing, aren't they?
Yeah.
We're looking at something like a 90% complete cure rate, often within just a week.
It's astonishing, truly, and it forces us to rethink how we talk about these microbes. You know, traditionally we called them commensals, like they just live alongside us, not really harming or helping.
Yeah, that term feels completely outdated now.
It absolutely is. FMT's success proves they aren't just passive roommates. They're essential partners. They form a barrier, they're a chemical factory, they process nutrients for us, they're critical.
Which really sets the stage for this deep dive, doesn't it? We need to grapple with this complex microbe-human relationship and why science is now pushing us to see ourselves as holobionts.
Right, a holobiont. That's the term for the host, us, and all our associated microbes living together and crucially evolving together. It's a concept that really gained traction after Joshua Lederberg coined the term microbiome back in 2001.
Let's talk scale for a second because it's hard to wrap your head around. You carry something like 10 to the power of 14 microbial cells. That's roughly 3 times more microbial cells than your own human cells.
The cell count is impressive, but it's the functional difference, the genetics, that's the real game changer.
Go on.
Our human genome. It gives us maybe 22,000 protein encoding genes, give or take. But sequence all the microbes living on and in you. Their collective genes add up to over 8 million.
8 million?
Yeah, it's like we're borrowing this incredibly specialized operating system, hundreds of times larger and more complex than our own DNA blueprint, to do jobs our own genes just don't cover.
And the Human Microbiome Project showed us that while these functions are vital, there isn't one single healthy microbiome blueprint, right? Everyone's is unique.
Totally unique, shaped by your age, your gender, diet, genetics, where you live. Everything plays a role.
But there are some common threads.
Oh, definitely. Across pretty much all humans, 5 major bacterial phyla tend to dominate. Actinobacteria, Bacteroides, Firmicutes, Fusobacteria, and Proteobacteria. The phylum level is limited, but within those, the species diversity is enormous. You know, the average adult gut probably hosts between 500 and 1,000 different species.
So how does this incredibly complex system even get started? Let's trace it back to birth. The way you're born actually matters a lot, doesn't it?
It's the first major seeding event. Babies born vaginally get colonized mainly by microbes from the birth canal. C-section babies, on the other hand, tend to pick up microbes more typical of skin environments, from caretakers, the hospital.
And this early start, especially specific bacteria in the infant gut, is critical for immune development.
Absolutely critical. We see this really clearly with bifidobacteria in breastfed infants. These aren't just passive bystanders. They actually synthesize growth factors the baby needs. And they have these unique surface proteins that let them grab onto and use specific sugars found in human milk, these things called glycans.
And then they ferment them.
Exactly. They ferment those sugars, producing acetate and lactate. These acids lower the pH in the gut.
Making it harder for bad bugs to grow.
Precisely. It naturally limits pathogens. And studies have even linked the presence of these bifidobacteria directly to a stronger, more robust immune response when infants get their early vaccinations. It's an immediate mutual benefit. High stakes.
Okay, so moving beyond infancy into adulthood, we start seeing distinct microbial communities in different body sites, these niches.
Yeah, the general rule is
Right.
internal tissues, brain, blood, cerebrospinal fluid, those should be sterile, microbe-free, but surfaces, skin, mucous membranes, the gut, heavily colonized.
Let's start with the skin. It seems like a tough place to live. Kind of dry, salty, slightly acidic.
It is challenging. And you get different niches even on the skin. You have dry areas, moist areas like armpits, and then oily, sebaceous sites.
Like the forehead.
Right, those oily sites are dominated by Cutibacterium acnes, formerly Propionibacterium acnes. These guys are really good at breaking down the lipids, the oils that our skin produces.
And that breakdown product.
Turns into volatile, unsaturated fatty acids. And here's a fun fact. Those fatty acids are a major source of what we perceive as body odor.
Huh. Okay, moving inwards. The respiratory tract.
Well, the upper respiratory tract, nose, throat, is quite diverse. The lower respiratory tract, the lungs, was actually thought to be sterile for a long time.
But not anymore.
No, we now know that's wrong. The lungs seem to function more like a revolving door. Microbes get inhaled, hang around for a bit, and then get cleared out. It's a transient population.
And the mouth. That changes early on too, right? With teeth.
Big time. Before teeth erupt, it's mostly aerobic bacteria. But once teeth come in, you get these spaces between the teeth and gums, low oxygen environments. That favors anaerobic species.
Like the ones involved in cavities.
Exactly. Organisms like Streptococcus mutants thrive there. They have ways to stick to surfaces and produce this sticky goo, a glycocalyx. That's the foundation of dental plaque, which can lead to cavities and gum disease.
Okay, then we hit the GI tract powerhouse. The stomach must be tough, incredibly acidic.
Extremely. pH 2 to 3, it's a major barrier, kills off most microbes that are just passing through. But then you get to the large intestine.
And that's where the party is.
That's the metropolis. Probably the densest microbial ecosystem on Earth. We're talking something like 10 to the power of 12 cells. That's a trillion per gram of intestinal content. It's packed.
Wow. So, given that everyone's mix of species is so different, how do scientists compare microbiomes?
They focus less on the specific names of the bacteria and more on what they do. The idea of a functional core microbiome. Meaning it doesn't matter so much if your gut has species A and mine has species B, as long as both species perform a similar essential function like breaking down a certain type of fiber.
And diet massively shapes this functional core.
Massively. A low-fiber, high-fat diet will select for microbes with a totally different set of functional capabilities compared to a high-fiber plant-based diet. You are literally training your microbial community with every meal.
We should also quickly mention the female genital tract.
Yeah.
That has its own specific protective community.
Very important, yes. Dominated by acid-tolerant lactobacilli, sometimes called Dutterlein's bacillus, they ferment glycogen, producing lactic acid.
Keeping the pH low.
Right, down around 4.4 to 4.6. That acidic environment is really crucial for preventing colonization by potential pathogens.
Okay, let's dig into those core functions more deeply. Metabolism and weight seem like a huge area. You mentioned germ-free mice earlier.
Yes, GF mice. Mice raised in completely sterile environments, no microbes at all. The experiments with them were eye-opening.
How so?
Well, GF mice actually eat more food than regular conventional mice, but they gain about 50% less weight on the exact same diet.
So the microbes are helping extract more calories.
That's exactly what it showed. Our gut microbiota is actively breaking down complex things in our diet. Things we can't digest ourselves, like certain fibers into products that our body can absorb and use for energy. They significantly contribute to our total calorie uptake.
And they proved causality with those transplant experiments.
They did. Took gut microbes from obese conventional mice, transferred them into lean GF mice, and guess what?
The lean mice got fat.
They gained weight, became obese, even though their diet and exercise didn't change. It demonstrated clearly that the gut flora itself can transmit an obesity trait. It's causal.
And the molecules driving this, the key players, are the short-chain fatty acids, SCFAs.
SCFAs are central. We absolutely rely on our gut microbes to ferment dietary fiber into primarily butyrate, propionate, and acetate. We can't do it ourselves.
Tell us about butyrate. Sounds important.
It's fascinating. First off, it's the main fuel source for the cells lining the colonocytes. They prefer butyrate over other energy sources like glucose.
Okay.
But beyond that, butyrate actually ramps up mitochondrial activity in those cells. It essentially makes them burn more energy, oxidizing carbon substrates to CO2. The source material described it almost like the calories are being vaporized right there in the gut lining.
Wow. What about the others? Propionate.
Propionate mainly travels to the liver. There, it seems to have a couple of roles. It can inhibit the synthesis of cholesterol, and it might also play a role in signaling satiety, suppressing hunger.
And acetate.
Acetate is the simplest SCFA. It gets absorbed easily into the bloodstream and can be used throughout the body, including as a building block for making lipids, like fats. So it's potentially a bit obesogenic, contributing to fat storage.
And obese individuals tend to have microbiomes that produce more acetate.
That's often what's observed, yes. A shift in the balance of these SCFAs seems to be linked to metabolic state.
Okay. Beyond metabolism, the microbiome is like our first line of immune defense, this colonization resistance idea.
Exactly. Your established normal microbiota actively prevents incoming pathogens from gaining a foothold. They protect their turf.
How do they do that? Directly attacking invaders.
Sometimes, yes. Bacteria can produce toxic peptides, things called bacteriocins or colicins, that specifically target and kill closely related competing bacterial strains.
But there are indirect ways, too.
Definitely. Microbial products, like those SCFAs we just discussed, or bits of bacterial cell walls, can trigger our own gut lining cells to produce antimicrobial peptides chemicals that kill bacteria.
And you mentioned butyrate having another immune role, something about oxygen.
Right. Butyrate also stimulates those colon cells to consume more oxygen. This lowers the overall oxygen level right near the gut surface, in the lumen.
And that favors.
That strongly favors the beneficial, strict anaerobes, the bacteria that thrive without oxygen. And it makes it harder for facultative pathogens, like certain E. coli or salmonella strains that can use oxygen, to compete and grow.
So the immune system itself actually needs the microbiome to develop properly. Those GF mice again.
Yes, the GF mouse model was key here too. They have really underdeveloped immune tissues. Their spleen, thymus, lymph nodes are smaller, less developed, and they produce very low levels of those crucial antimicrobial peptides.
So the microbes are sending signals.
Constantly. It's essential. Microbial signals, whether it's SCFAs or specific molecules like bits of peptidoglycan or LPS, that our immune cells recognize using toll-like receptors, TLRs. These signals are required for the proper maturation and function of immune cells like neutrophils and macrophages, not just in the gut, but throughout the entire body.
Okay, this leads us to maybe one of the most intriguing areas, the gut-brain axis, the GBA.
Yeah, it's a super hot area of research. And again, GF mice provided early clues. They showed distinct behavioral differences compared to normal mice, things like increased anxiety, being less sociable.
And you can change that just by giving them bacteria.
You can. Administering certain bacteria or even just specific microbial products like butyrate can actually normalize some of these behaviors in GF mice. It's pretty remarkable.
How does that communication work? How does the gut talk to the brain?
It seems to be multi-pronged. There isn't just one pathway. One major route is through immune modulation.
Okay.
Microbial products, especially if the gut barrier is compromised, like that LPS we mentioned, can trigger inflammation. The inflammatory messengers, cytokines, can travel through the blood to the brain.
And affect brain function.
Yes, they can alter how neurons and microglia, the brain's immune cells, function. This is linked to things like cognitive changes, fatigue, the sickness behaviors you feel when you're ill.
What's another pathway?
There's a direct neural pathway. Our gut has its own complex nervous system, the enteric nervous system, sometimes called the second brain. Gut microbes can stimulate nerve endings in the gut wall.
And that signal goes up?
That signal travels directly up to the central nervous system, primarily via the vagus nerve. It's a direct communication line.
Wow. And there's a third mechanism, something about physical protection.
Right. Protection of the blood-brain barrier, the BBB. This is the highly selective barrier that protects the brain from harmful substances in the blood. SCFAs, particularly butyrate again, seem crucial for maintaining the integrity of the tight junctions between the cells that form this barrier.
Keeping it sealed tight.
Exactly. And guess what? GF mice, lacking these microbial SCFAs, have a more permeable, leakier BBB. It shows the microbes contribute to the physical protection of the brain.
It's incredible how interconnected it all is. Which brings us, unfortunately, to the flip side, dysbiosis, when things go wrong.
Yes, Dysbiosis, that's the term for when the microbial community is altered, unbalanced in a way that contributes to disease. And often the underlying issue is chronic, low-level inflammation.
Like with metabolic syndrome.
That's a classic example. Metabolic syndrome involves a cluster of issues: high blood pressure, high triglycerides, insulin resistance. It's strongly linked to something called metabolic endotoxemia.
Endotoxemia. That sounds like toxins in the blood.
Precisely. It often starts with diet. A high-fat diet can reduce microbial diversity and damage the gut lining, leading to that leaky gut.
Right, the loosen, tight junctions.
So now, lipopolysaccharide LPS, that component from the outer membrane of gram-negative bacteria, can leak across the gut barrier into the bloodstream more easily.
Even small amounts.
Even low levels. But chronically, this circulating LPS triggers a persistent low-grade inflammation throughout the body. And that inflammation is thought to be a major driver promoting insulin resistance and eventually cardiovascular disease.
And diet plays a role in cardiovascular disease risk through other microbial pathways too. Something about meat.
Yes, another fascinating link. Compare fiber-rich diets, which feed beneficial microbes, to diets high in red meat and saturated fat. Certain gut microbes thrive on compounds found in red meat, like L-carnitine, and also phosphatidylcholine found in fatty foods.
What do they do with those?
They metabolize them into a compound called trimethylamine, or TMA. Our liver then takes that TMA and converts it into TMAO, trimethylamine and oxide.
And TMAO is bad news.
TMAO has been shown to accelerate atherosclerosis, the buildup of plaques in arteries. It's a direct link from specific dietary components through microbial metabolism to increased heart disease risk.
Is this different in vegetarians?
Significantly, studies show that long-term vegetarians or vegans have much lower levels of the microbes that produce TMA, and consequently, they produce very little TMAO even if you give them a carnitine challenge. Their microbiome is different.
We also have to touch on the connection to cancer. You mentioned about 20% of malignancies might be linked to microbes.
That's the estimate, and it can happen in a couple of ways. There's direct carcinogenesis,
Meaning the microbe itself causes cancer.
or produces something that directly damages DNA or messes with cell growth. Good examples are certain E. coli strains that produce a toxin called colobactin, which damages DNA. Or think about Helicobacter pylori in the stomach. Some strains inject a protein called Kg directly into stomach lining cells, and Kg dysregulates cell growth pathways, increasing stomach cancer risk.
But there's also an indirect link back to inflammation.
Exactly. That chronic low-level inflammation we talked about, driven by things like obesity, metabolic endotoxemia, dysbiosis, that itself creates an environment that promotes cancer development. It increases the risk for various cancers, including colorectal, liver, and kidney cancer, among others.
Given all this, the good and the bad manipulating the microbiome therapeutically seems like the obvious next step. Probiotics are huge now.
Huge. It's a massive industry, something like US $30 billion. The official FAO-WHO definition is important. Live microorganisms, which, when administered in adequate amounts, confer a health benefit to the host.
But it's a bit Wild West in terms of regulation, isn't it? At least for supplements.
For supplements, yes. In the US, the FDA doesn't regulate them nearly as strictly as drugs. But if you're talking about using live microbes as a defined treatment for a specific disease, like a standardized, quality-controlled FMT preparation for C. diff that gets regulated like a drug, requiring rigorous clinical trials for safety and efficacy.
That makes sense. And what about symbiotics? I've heard that term.
Right, a symbiotic is simply a product that combines a probiotic, the live beneficial microbe, with a prebiotic.
And a prebiotic is?
A prebiotic is basically food for the probiotic. It's usually a type of fiber or compound that selectively encourages the growth and activity of the beneficial microbes you're introducing, or the ones already there. The idea is to give the good guys a boost.
This isn't just for human health either, right? Agriculture's using this too.
Oh, absolutely. There's some really promising applications there, potentially reducing the need for antibiotics in livestock. For example, adding certain strains of Lactobacillus acidophilus to cattle feed has been shown to reduce the amount of pathogenic E. coli 0157H7 the cattle carry and shed, sometimes by up to 60%.
Wow, that's significant.
It is. Similar approaches are being used in poultry farming to reduce salmonella. It's a practical way to leverage microbial interactions for better health and safety.
So wrapping this all up, this deep dive really forces you to confront this idea.
Yeah.
Your microbiome isn't just there. It's functionally like a whole other organ we didn't fully appreciate until recently.
Absolutely. From the shock and effectiveness of FMT to the way SCFAs like butyrate manage energy to that intricate communication along the gut-brain axis, our microbial partners are just indispensable for our health, for maintaining balance, for homeostasis.
But there's a catch, isn't there? A challenge in making positive changes stick. We know that even if someone loses weight or drastically changes their diet, the microbiome doesn't always snap back immediately. It can lag behind.
That's a critical point. This persistence of dysbiosis, or the memory of the previous unhealthy state in the microbial community, can make it harder to maintain weight loss or health improvements. The microbiome might still be geared towards the old way of life.
So this leaves a really important question for you, the listener. If we know the microbial community can be resistant to change, and we know it's the functional core, what the microbes do, that really matters.
Then what specific knowledge, what behavioral strategies do you need not just to change your diet, but to actively manage and reset these microbial partners for the long haul? How do you ensure they maintain that beneficial functional core needed for lasting health and stability? Is diet alone enough, or is it a more continuous process of cultivating your inner ecosystem?
That's definitely something powerful to think about for your own health journey. Thank you so much for joining us on this deep dive into the fascinating world of the microbe-human ecosystem. We'll see you next time.